This case study is on OCD, obsessive compulsive disorder, specifically valence processing alterations in SAPAP3 knockout mice and human OCD. Valence processing is a factor to OCD because OCD is known to cause a lot of distress and anxiety with those who have it. And in this research paper, it demonstrates how the valence processing in SAPAP3 knockout mice are similar to behavior seen in OCD patients. Auditory fear conditioning and extinction were used to examine valence processing in the knockout mice, and a set of behavioral tests were used to examine the valence processing in the OCD patients. The results do in fact show that valence abnormalities in the knockout mice are similar to those seen in OCD. This is just some background on what valence processing is and what the SAPAP3 gene is. So the neural circuits that process valence are significant when it comes to behavioral responses to people, objects, and events. So a dysfunction in those systems can play a role in OCD. Now, valence processing is the degree at which something is pleasurable. So a positive valence involves emotions of happiness, enthusiasm, and excitement, and negative valence is the opposite. So it involves emotions of anxiety, fear, and sadness. The frontal striatal circuits help to mediate cognitive, motor, and behavioral functions in the brain. So the loss of the SAPAP3 gene causes impaired frontal striatal synaptic transmission, which contribute to OCD-like symptoms in the mice, such as increased anxiety and compulsive grooming behavior, which would be a negative valence. So the SAPAP3 knockout mice show consistent trends towards enhanced negative valence processing and impaired positive valence processing. And the human OCD patients show enhanced responsivity to negative valence images and decreased responsivity to positive valence images. For the methods and materials, male and female SAPAP3 wild type and knockout mice were used for the experiment. To test the valence processing in the mice, an auditory fear conditioning and extinction paradigm was used. The mice were placed into a conditioning cage where they would hear an auditory tone followed by a brief shock. When this happens, the mice will freeze. So on the fear conditioning day, mice were allowed to roam the inside of the chamber for 2 minutes, which is marked by, well, this is called the pre-tone period. So following this, they were exposed to a 4-tone shock pairing. After conditioning, the mice returned to their home cage and were left undisturbed until the following day. On the fear extinction days, mice were allowed to roam the chamber for 2 minutes, which is the pre-tone period, and they were exposed to 4 tones with no shock delivery. This experiment demonstrates how the mice react when an adverse stimuli, the shock, is introduced and then removed. Differences in percent freezing during the pre-tone period and during the tone period between the wild type and knockout mice were measured with ANOVA and SIDX correction for comparison. And ANOVA is just a test for analysis of variance, and SIDX correction is used to show multiple comparisons. So just to sum up the experiment, on the fear conditioning days, wild type and knockout mice are placed into the cage where they hear a tone, and then a shock comes right after it. So the mice will associate this tone with the shock. And on the fear extinction days, the goal is for the mice to extinguish the fear of that tone, so they will just hear the tone without a shock. To test the valence processing in human OCD patients, an explicit and implicit emotional processing task was conducted. Tests were completed in a single undistracted sitting of approximately 45 minutes. During the explicit part, participants had to select as quickly and accurately as possible the correct label from a list of emotional expression labels on a computer. 8 images of each expression type were randomly presented for 2 seconds. And I included the chart to give an idea of what exactly the participants saw. So there's the happy expression and that fearful expression. After this, there was an interval of 20 minutes where participants completed an unrelated cognitive task. For the implicit part of the task, the 48 previously seen faces were paired with 48 unseen faces that were matched by sex, emotional expression, and other presentation parameters. From each presented image pair, participants were asked to select again as quickly and accurately as possible the image they had previously seen. Basically, it was a face recall task for the implicit part. In this analysis, the focus was on explicit and implicit processing of expression of fear and happiness. Z-scores were calculated and reflected performance based on accuracy and reaction time. The results from the fear conditioning and extinction paradigm shows that knockout mice had approximately 75% freezing during tone 20 compared to wild type at around 30%, which that's a huge difference. Wild type had a significant decrease in their freezing tone from early to late extinction, and the knockout mice had shown no difference in their freezing tone from early to late extinction. This suggests their impaired ability to extinguish fear. The SAPAP3 knockout mice showed evidence of enhanced fear learning and impaired fear extinction, which contributes to their enhanced sensitivity to negative valence. The results from the explicit and implicit emotion processing task show us that OCD participants were significantly less accurate for face recall by fearful expression, but were more accurate at identifying fearful expressions than identifying happy expressions. And that demonstrates how OCD patients respond more effectively to negative valence stimuli than positive with greater accuracy and speed. They also had a high mean Z-score of about 0.5 for fearful expressions compared to negative 0.5 for happy expressions. The data and the evidence, they show that OCD may indeed be characterized by key valence processing alterations. And just to mention again, the lack of the SAPP3 gene causes OCD-like symptoms in the knockout mice because they have increased or enhanced negative valence processing. And for the human OCD, they are more accurate at identifying fearful expressions with greater accuracy and reaction time, which demonstrates their negative valence processing. Just to also add, I think it's really interesting to know that mice can have similar mental health issues as humans and that they can actually suffer from OCD. And I actually like any research article that deals with mice since they're so similar to humans, even though I kind of feel bad that they had to be shocked like that. But I think it'll help with future research on the disorder. So it's for a good cause, I guess, I suppose. Okay, so for limitations and future directions, this work supports 2 new approaches for the study of valence processing abnormalities across the spectrum in OCD that include the SAPAP3 knockout mice and the emotional phase paradigm. The limitation would be the lack of testing of a healthy control group along with the patients with OCD. Having a control group would actually help to effectively compare the valence processing in those with OCD and those who do not have it. Future work should focus on using similar tasks and experiments between animal and human studies. Tasks which combine trials to obtain reward and trials to avoid punishment through either performing or withholding an action may be better for increasing the translational potential of findings between human and animal studies.